from the Institute of Experimental Botany in Prague, and I'm from the lab of pathological plant physiology. So I'm a plant physiologist and plant pathologist, and I would like to talk today about how we implement the imaging facility and imaging facility techniques into our more physiological questions, and something about the challenges that we face with it. So a bit for context, which was already shown at this meeting, we work with the question of barley production, which is very important for Czech Republic and for EU in general. And we bring the barleys, we don't do field work, we do experimental work in the lab in controlled conditions, and we study the background of barley resistance to pathogens. So one of the pathogens that is currently attacking a lot the productivity of barley is their Blumeria graminis, the biotrophic pathogen that is quite widespread, and the main measures how to fight it is to create the resistant varieties. So when the barley suffers from this pathogen, you can see that on their barley leaves, you can see white stripes. And these are the spores of the pathogen, which is spread around the field, and it is spread by air. So it's really hard to control, to quarantine this disease. The life cycle of this pathogen is rather complex. So it has complex cycle that includes propagation, sexual propagation, and it's connected with their wintering and their longer circulation in the field. And another cycle that is important more for the spreading within the field is the cycle of asexual propagation. So in this case, the spores fall on top of the leaf, and then they start to penetrate, and then they get within the tissue, spread, and in the end produce the next spores that will be then spread by the air. So the whole cycle in this case takes about 24 hours, and it makes it quite convenient to study in the lab. And this is quite the question that is important for the epidemiology of the disease within one vegetative year. So what we're interested in is what happens at the cellular level. So we study the process of spore falling on the leaf and their initial stages of the interaction between pathogen and the plant cells. And usually there are several anatomical structures that can be easily distinguished. Is there germinating spore, which has the first germinating tube, the primary tube, and then the secondary hyphae that forms a presorium, which is the end of the tube, putting pressure on plant cells and also producing enzymes to modify plant cell until another hyphae can penetrate inside and spread. So when we work with it in the lab, we have their experimental setup on how to apply the spores on plants to make it the most reproducible and the most homogeneous possible. So we use so-called inoculation tower, which is basically just the empty metal tower. And we put the barley leaves on their on the horizontal surface, because normally they stand vertically and it's hard to apply something on them reproducibly. So we just bind them to the horizontal surfaces and place them barley plants or another experimental plants at the bottom of the tower, then take some leaves from the already infected plants which have a lot of spores and just apply them through the tube with the pressure of air. So in this case, gravity helps us, and we use always their glasses to control their level of infection. Why I'm talking of that in detail? So to show you that the infection is very heterogeneous, and we are trying really hard that if we want to assess the physiology of the disease, we need to ensure that the spores are distributed as homogeneously as possible. So in this case, in case of barley, we usually use double staining, which we can stain the hyphae. There are classical long-nose stains to visualize the hyphae within the tissues. Like here with simple trypan blue, we can observe the conidia being formed and the hyphae being spread within the tissues. And when we want to focus more on the plant side of the response, we usually visualize the production of secondary metabolites by specific staining. And the classical one is the aniline blue staining for colors, which actually also stains their cells of the pathogen, but mostly the colors deposition within their cell walls of the neighboring infecting cells. So, but when it comes to how do people evaluate the disease and what is the golden standard to study barley interactions and the barley resistance, it is still comes, like even nowadays, it comes to the method from the 70s, which is just the disease scaling. So it's certain things we can visualize better with the imaging, but then when it comes to actually putting numbers on it, we're coming back to human evaluation just by eye, evaluating the percentage of the leaf which has some kind of disease symptoms. And this has been like golden standard till now, but here you can see there different barley lines with different production of lignin and their secondary metabolites. And you can see that what we scale is their disease tissue. And by that, we can see that some of them look healthier, some of them look worse. So it's just like to show you the step back that we have to take when it comes to disease evaluation. Not everything can be yet automatized. Not everything can be outsourced to AI and to like learning machines. We have to go back and just look at it. So what we can also do by applying a little bit more imaging when we are interested on plant responses, we do also different staining and to visualize the plant immunity activation, like just staining the hydrogen peroxide. You can see that when the infection is being stopped, the immunity is functional, then we detect the ROS production, and the cells are just dying without letting the pathogen spread. And when the immunity is non-functional, in the susceptible lines, which show yellowing in the end, we can detect no hydrogen peroxide production, but then the hyphae get much broader and like you can see much more trypan staining on the leaves. And this is something that we can only see when we do staining, and we cannot evaluate by just looking at it. So how we are trying to solve the barley resistance. To solve the barley resistance to Blumeria, we use their plant which is naturally non-resistant, the non-host Arabidopsis. And we aim to use the mechanisms how the Arabidopsis is non-host to put this mechanism to protect barley, which is the susceptible host. So we use the non-host system. We apply their pathogen spores on Arabidopsis surfaces with the same inoculation tower approach. And in this case, the resistance in Arabidopsis is based on papilla formation, which is the very specific structure, like reinforcements of cell wall around the plasma membrane that help to protect against invasion. And in case of barley, this structure also forms, but it's mostly unsuccessful. And in case of Arabidopsis, it's mostly successful. So with the same staining, we can visualize their colors and their hyphae by the double staining. And then if we look a bit further away, we usually have a look and measure the just area of the callus deposition within the leaf. And there is a certain threshold to which the callus is functioning. And if it's too much, it also indicates some troubles in the defense. And we can also monitor the callus deposition more locally and their accumulation of their specific disease-associated proteins in the papilla. So what we also noticed while working with the system is as their infection is heterogeneous, so is the immunity. Like for us being able to detect just the activation of defense genes by their gas fusions or the venous fusion, you can see that their diseases, that immunity is progressing the same like 1 and 2 days after infection. But also we can see that activation of defenses is heterogenic, not only within the leaf in their like horizontal scale, but also within the upper and lower sides of the leaf. So what we're dealing with is their activation of immunity really at the level of single cells and neighboring cells. So the things that we have to also address is cell-to-cell communication that is involved in stopping their disease response. But then when it comes to how do you evaluate the success of their barley penetration or not, in Arabidopsis, we have to use, again, some kind of human-based approach. And we usually assess like about 100 papillas of each genotype, because each interaction of spore with plants is considered a very individual case. So what we usually do, we just observe as many papillas as we can. And the successful situation is when you can see the papilla with certain callus number, callus accumulation, and a certain structure at the bright field, which indicates a little thickening of their cell around around the interaction case. But this is a successful defense case, and the defense failure has different phases. And what we usually evaluate is their excessive callus deposition or granulated cytoplasm or local cell death or haustoria inflammation. So we are still back to the situation when we have to categorize responses, and it is only done by humans observing many papillae under the microscope and putting categories in the numbers and measuring the probabilities. But what we are doing next, we are studying the successful case of interaction, the papilla, and looking into it a little bit deeper. And to do so, we apply several dyes. For example, FM464 stains nicely plasma membrane, but also the spores. And if we overlap it with aniline blue staining, then we can focus on the callus deposition exactly at the places of penetration, and then analyze the shape of the papilla, the intensity, and the speed of how it forms after the infection. So the challenges that are often overlooked in the literature is that if we look at the papilla at the same GFP positive staining, it is very autofluorescent. So you can see that this is like no staining. It is wild-type plants, no GFP present. But if we do if GFP fluorescence testing, we can see the papillas anyway. So this is just often not being presented in literature, and it's really difficult to evaluate, like, what are we looking at? Are we looking at GFP, or are we looking at just cell wall thickening or reflecting properties of this papilla? So we were expected that this may be the callus, but then we took the mutant which cannot make callus, which is deficient in callus synthase, and we still saw certain autofluorescence. So we have thought a lot about how to deal with it, and for now we have decided that we are not doing aniline blue staining anymore. We just ordered another alternative fluorophore, which is supposed to have a more narrow spectrum. We're applying a mixing of emission wavelengths. We do stronger background subtraction, but we still have to do a lot of controls to make sure that the fluorescence we observe is attributed to the GFP and not to the just cell wall properties. But with this new aniline blue dye, which is our alternative just aniline blue fluorophore, and with the double staining with the plasma membrane, we now can do quite nice reconstruction of callus or callus-associated compound deposition in papilla, and we can see that it is quite heterogenic. But why are we looking at this question-wise is we want to see how this callus gets there. And to see how the callus gets there and makes the papillar reinforcement, we're interested in how much the plasma membrane surface in this place of interaction is changing within the infection. So what is known by now is that certain phospholipids like phosphatidic acid are enriched in the papilla formation, and they help to recruit other lipid-modifying enzymes like PLDs, and then they can target vesicles to the place where they're needed. What we were interested in is what about the phosphoinositides and other polar-rich, polar-charged membrane lipids, which are involved in trafficking in different contexts, but have never been really studied in this non-host interaction system. So we took their lipid sensors developed in, I think, French-affiliated bioimaging unit. So we used the lipid sensors which mark the enrichment in phosphoinositide phosphate, and we saw that indeed there is enrichment also in phosphoinositide 1-phosphate in the papilla. And this enrichment really nicely colocalizes with the callus deposition. So we can say that the membrane needs to change its properties to recruit more callus synthases and to produce more callus. This is our hypothesis for now. And if we look at the second, another minor changed phospholipid, we tried to do it with the same setup with a certain sensor. And we did saw it as well in the papilla, but then another challenge was that the sensor was only expressed in cotyledons, but not in true leaves. So we had to redesign our experiment into working with the younger plants, but saying to ourselves that papilla is still papilla. So what we know for now, that there is a certain enrichment of phosphoinositides and plasma membrane at the interaction of growing hyphae with the plant cells. And what happens, what is the biological sense of this, that if we take the mutant which cannot produce these phospholipids, we do see a strange callus deposition pattern. But instead of seeing less of callus, we see much more of it, and it starts to be deposited not only in the papilla places, but like scattered across the plant cells. So there's just like excess of callus, which is not going where it should be, but instead it's spread everywhere. And when we look deeper on how the callus is deposited in papilla, if still deposited, it is really scattered, messy, and what is the most important for our phytopathological case, that these plants, which cannot have the phosphoinositides at the right place, they also suffer much more from the pathogen penetration. So we are putting it all together to make sense how the membrane changes are really important for the proper forming of the papilla, which is the major mean of protection for non-adapted pathogens. So from my case, we had a lot of fun, and we still have a lot of fun imaging the papillas, because every papilla is very different, and we cannot really outsource it all to automatic learning. We have to look at them ourselves and choose the proper ones. But what I wanted to show here is mostly that the interaction of fungal pathogens are a bit more complex than just bacteria, because each spore, each hyphae has a bit different impact on plant cells. And we have to use complementary techniques to really evaluate the immune response. We can not only rely on imaging of fungal side, on imaging of plant side to evaluate the outcome of the infection. And we plan to continue going on to study the phosphoinositide turnover, which is like important for basal resistance. And for this, I have to thank to our imaging facility and people involved in this work, which is a pathological plant physiology group in collaboration with structural cell biology group, and with our imaging facility presented here by Katka Malinska.
My name is Tetiana Malinska, and I would like to very briefly introduce the Prague node of advanced light and electron microscopy. So all tourist guides advertise Prague as a beautiful historical city with an excellent beer. So why not to connect this with the work or with imaging at the Prague node of advanced light and electron microscopy, or with visiting one of our many workshops organized here. Maybe we can gradually create a new touristic landmark here in Prague. So who are we? The Prague node consists of 6 facilities, which are located at 5 different institutes in Prague or in South Bohemia. Prague node is coordinated from the Institute of Molecular Genetics. And we have one facility dedicated directly to the plant imaging, which is the Institute of Experimental Botany here in the north part of Prague. So what are we able to offer? From a lot of movement in this slide, it's clear that we really love non-invasive in vivo microscopy with close to physiological conditions. We offer a plant-dedicated imaging solutions at a very different time and spatial resolution, which starts from centimeters and a week of imaging over typically overnight imaging on our vertical stage confocal microscope. Very popular are drug treatments done in perfusion chamber, which are done usually in the range of minutes, or very fast imaging acquired with a scan resolution and let's say 10 on hundreds of milliseconds time resolution. So for those of you who needs higher resolution, the Prague node also offers super resolution and electron microscopy oriented facilities. One example might be the Charles University facility in Biosef Institute, which is headed by Aleš Benda, and I saw here also David. Aleš is the scientific leader of our Prague node, and this is a small example of one multi-facility project which started here at our institute by simple vertical stage confocal microscopy done by Adriana Jelinkova, and then she continued at the Biosef facility further with FIPSEM to localize some internal PIN1 structures. So, because it's quite late, I would like to thank you for your attention. I would like to invite all of you to Prague. And mainly, I would like to thank Euro-BioImaging for organizing this outstanding afternoon, which I believe everybody of us enjoyed very much. Thank you.